Hello, I am Wonjun Erickson, one of the authors of this paper. The study is about emulating a movement disorder called childhood hypertonic dystonia and cerebral palsy in a synthetic neuromorphic system running in hardware. We are interested in understanding what abnormality in our neural system is responsible for characteristic features of dystonia. So here we have a simplistic neural pathway for joint movement. A shorter loop for the spinal reflex and a longer loop for the transcortical pathway. We want to model this system and later introduce hypotheses for the pathology of dystonia to find a sufficient mechanism for the movement disorder. Here is a schematic that represents the implemented circuit. And a rotating arm in the left is pulled by two antagonist muscles. In order to create a biorealistic testbed, thousands of spiking neurons and virtual spindle, muscle, synapses, and electromyograms are implemented in FPGA chip. If you probe the output of the neuron using an oscilloscope, you can see the spike trains in response to stretch of the muscle. We also have a population of neurons with different soma sizes acting together in ensemble to ensure the size principle in the muscle activation. So let me show you the setup. The freely rotating virtual arm in the right window represents the elbow joint. This is a plant that is connected to the neural controller residing in the 6 FPGAs in the bottom right. If you virtually perturb the joint in some way, you see the response of the reflex taking place. For model validation, we show here the multi-scale data for the stretch and hold test. You can see the neuron raster with the correct physiological delays in sensory and motor neuron. Muscle force and EMG is also generated. Now that we have a non-impaired system, we can test hypotheses for the possible mechanism for the hypertonic dystonia. From previous studies, hypertonia may be due to increased activity in the long-latency reflex. So we modeled two possibilities of hyperactive long-latency stretch reflex by either creating a tonic involuntary drive to a cortex or increasing the synaptic gain in cortical neurons. We compare the results of our model with human data whenever there exists any comparable one. The top panel shows about 80 seconds of passive back and forth stretch applied to the child with hypertonic arm dystonia in Van Doornik's study in 2009. The plot immediately below shows a biceps EMG response to the stretch. We see the biceps EMG modulates with the angle. In the emulation, we use the same waveform as in the original study to stretch the emulated system. Our example result shows that in both tonic and high gain models, the EMG activity modulated with the joint angles, similarly to the original human experiment. The models of dystonia are validated based on their ability to qualitatively explain human data. So we calculated the phase angle between the model-generated EMG and the joint angle using the same approach in the human experiment and compared with the 6 available human data. The humans showed a range of phase angles between -30 and -107 degrees. We validated our models of dystonia by testing whether the phase angle in human can be achieved in emulation. We scanned the representative variables in both tonic and high gain models and discovered the range of possible phase angles in emulation. It turns out the tonic model produces phase angle from -32 to -53 degrees, which explains only 1 out of 6 patients, as shown in dark gray in the left, whereas high gain model produced phase angle from -29 to -147 degrees, as in the dark gray area on the right, which covers the phase angle of all 6 subjects. The results suggest that in terms of phase angle, the high gain model can explain more data than the tonic model, although more human data are required to distinguish between the two models. This was a short overview and introduction of one of the many results. For the rest of the study, please read the paper.